Hi everyone. So I'm sorry I couldn't be here today. However, I did want to give you a little bit of an overview about cell organelles. I know that you have already heard from your classmates about these different organelles. However, I wanted to give you a little bit more in depth today. And I also wanted to give you some interactive portions as well. So just like before, I would like you to take notes on these. A lot of the notes you already have, so I would like you to add to those notes if you would like to. There are going to be some discussion questions that go along with some of the 3D modelings that you guys are going to see in the Nearpod. So what I need you to do is first take out whatever way that you're going to be taking these type of notes. And second, I need you to log on to nearpod.com. And I need you to go ahead and put in this code. So we have AFQIR. So I want you to go ahead and put that code in for me, and let's get started. So, cytoplasm. Cytoplasm is extremely simple. Don't think too hard on it. It is this semi-fluid material called cytosol, and it's found inside the cell membrane, and then it surrounds all the organelles. So probably the best way to explain it, if there is an explanation to be made, is think of a swimming pool and the water and all the pool noodles and other things are floating in it. The water, that's the cytoplasm. Cytoplasm literally just keeps everything inside the cell. It's so that it's not just floating in space, it's actually floating in this kind of situation. Now, semi-fluid. Make sure you understand what semi-fluid means. Semi means kind of or slightly or sometimes. So it's semi-fluid. Think of it, it's going to have the same consistency as Jello or jelly, something like that. And so, yeah, it's pretty simple. Moving something around. Okay, next thing is cytoskeleton. So cytoskeleton is the framework of a cell. It's going to be usually on the outside portion of the cell, and it's going to keep its shape. So there are two types of protein fibers that it's made of: microtubules, which are made of tubulin. These help shape and support the cell. Then there's microfilaments, which are made of actin, and these allow for cell movement and an intracellular or in-cell transport. That means moving one thing from one corner of the cell to the next one. That's pretty much all the information you need for that one. Next one, okay. Cell membrane is extremely important, but I'm going to tell you this right now, is that we are going to be going in extreme depth with cell membrane, but for this time being, this is really what you need to know about it. So, semi-permeable membrane, which means that if it's semi, that means it's sometimes, and permeable means it's allowing something to come in. So if you have a semi-permeable membrane, it means that sometimes it's letting things in or out, depending on what they are. At this time, you do not know what those things are. However, you will. Don't you worry. So, like I said before, it's going to surround the cytoplasm of the cell. It's really on the outside, like if you see in this image right here, it's the outside of the cell, and it's keeping everything in. So it helps make sure that the cell does not dry out. And pretty much the macromolecule that's the most important with the cell membrane is lipids. In animal cells, you can find the cell membrane. It's a flexible, thin outer layer. It's the outermost layer. And in plant cells, you're going to see it beneath the cell wall. And we're going to talk about the cell wall in just a little bit as well. So at this time, what I want you to do is you're going to go to Nearpod. And I have just an example, already up so that you can see what I'm looking at. And I want you to actually move the image from side to side. I want you to zoom in on it. And then I want you to let me know, what do you notice about these layers? So if I look over here, so you're going to see 3D cell membrane. Here. You're going to move on to the next one. So here's actually an extremely zoomed in version of a cell membrane. Now you have to understand, like if I was looking at a cell, this is what you would actually see. So this is only one little small section of the cell membrane that takes over the entire cell. So let's say like this is the cell, this is like the little dot or something like that. There's so much of the cell membrane. So this is if we were to look at it in a sectional view. So if you want to, you can actually zoom into this. Whoop, I went a little too far. So I want you to look at this, and you can play around with it as much as you want, or you can pause this if you feel like it. And I want you to be able to answer the question that I just had. So what do you notice about the layers? Take a minute, and you can pause this and come back. But we're going to go ahead and move on to our next one, which is centrioles. So, centrioles, they are cylindrical structures found in animal cells, only animal cells. Cylindrical just means that they're cylinder-shaped. And as many of you have described, they kind of look like Twizzlers. They are made of microtubules, and then they are involved in cell division. Now, here's something that I want you to realize, and I want you to add in, is that they only appear during division. So what I mean by that is when division's done, they actually disperse and they disappear into like just singular microtubules. And then when the cell is about to divide again, they come all together to make this nice cylinder shape, and then they help the cell divide. Next one, nucleus. There's a lot going on with the nucleus. So first things first, the nucleus is the cell's managing organelle. I like to pretty much call this the control center. So here's the thing with our nice control center. Think of the nucleus as the brain of a cell. That's pretty much what you need to know. So it contains most of the cell's genes or its DNA. The nucleus is actually surrounded by this wonderful nuclear envelope. And then the nuclear envelope contains these pores, these little bits of pores or openings that allow substances in or out. And then you have the nucleolus. So the nucleolus, which is this portion right here, and that actually is where ribosomes are made. So it's very important. You're now going to go ahead to that Nearpod again, and you're going to see a 3D image of a nucleus, which is pretty cool. So make sure you're looking at it, and I want you to let me know what the holes represent. So what do these holes represent? And actually, if you zoom in, there's a red ball in the middle of it. I want you to be able to try and figure out what exactly does that red ball represent. You're next going to go ahead and look at a 3D image of a DNA, RNA, ribosome. Looking at these images, so we have DNA here, RNA here, and ribosomes here. So looking at these images and thinking of this, why do you think RNA and ribosomes can leave the nucleus or through that nuclear envelope, those pores, but not DNA? Think a little bit, write it down, and come on back. All right, ribosomes. So ribosomes are the cell's protein factories. You can find them on the nuclear envelope, on the ER, but only the rough ER. That's what that little asterisk means. And also floating, just free floating in the cytoplasm. So they're a site for making protein or protein synthesis. And although they're not generally considered an organelle because they do not have that like a nice membrane around it. This is why prokaryotes can have them. And eukaryotes have them as well. This is why, because they're not a membrane-bound organelle. So, there you go. All right. Endoplasmic reticulum. It's real fun to say. We pretty much just say this is ER. Now, there's two different types of endoplasmic reticulum, and they are both extremely important. The rough ER, which has ribosomes in it, is involved in making and processing protein for export, which means that they're eventually going to be moved out of the area of the endoplasmic reticulum to other areas of the cell. The smooth ER, which has absolutely no ribosomes on it, is actually involved with producing lipids and then detoxifying of drugs and poisons that are in the cell. Now, the cell can have a lot of different poisons and drugs that come into it, both just through like natural ones that your body actually produces and needs to get rid of, and actually ones that you actually introduce into your body. That's what the smooth ER is for. That's why you have so many of them. For example, you're going to find a lot more larger and bigger smooth ERs in your liver than you are in like a muscle, things like that. So you can go ahead, actually, if you go to the Nearpod, you're going to find a 3D image of endoplasmic reticulum. Same thing as before, mess around with it, look a little bit, zoom in, things like that. And so tell me what you notice about the rough versus the smooth. All right, next is Golgi bodies. So Golgi bodies are fairly interesting. They are a stack of flattened sacs. That is like the best way to describe them. And they have three main functions. They modify and package proteins. They transport materials, and they produce lysosomes. And lysosomes are extremely, extremely, extremely important, and we're going to talk about those a little bit more. But first, I want you to go ahead, go to Nearpod, and I want you to be looking at our 3D image of a Golgi body. And I want you to go ahead and look. So as you can see, there's like these circles or, different things that are coming off the Golgi body. As you can see, I want you to try and let me know what you think these bulges are that are coming off of it. And then you can come on back to this. Okay, so lysosomes. So these are only found in animal cells. Please know that. So I guess I should probably make this a little bit bigger. But yes, these are only found in animal cells. So far we have seen centrioles and lysosomes, which are only found in animal cells. So lysosomes, you contain enzymes that break down organic molecules in old worn out cell parts. They're considered the cell's digestive system. Pretty much what that means is that if there's any food that comes in or anything like that, the lysosomes are in charge of breaking it all down so that it can actually be used for energy, or it can be, taken out of the cell itself, things like that. And you're going to go back to Nearpod, and you're going to go ahead and look at it. I don't have a question for this. It's kind of boring looking, to be completely honest. It's a lysosome. It's not really doing, it doesn't have to look special to have a really important function. Okay, vacuoles and vesicles. So these are membrane-bound sacs within the cell, and vacuoles are a type of vesicle. There are so many different types of vesicles. It's incredible. However, vacuole is just one example of those vesicles. I mean, there's so many different types of vesicles, and we could talk all day about those. However, right now, just vacuoles is what we're talking about. And they're extremely important because you find them in food, waste, and water. Now I want you to look at these pictures. So in plant cell, here is the vacuole. And then in our animal cell, we have these green. So if you look at these green dots, those are also the vesicles. What do you notice about the size of them and the number of them? Go ahead and write that down for me. So before I start into the next thing, I want to let you know that this is the end of part 1. And part 2 will be the next video that I want you to go ahead and look at and start. So again, this is the end of part 1, and go ahead and click on part 2, and you can go ahead and start that. All right, see you over there.